Okay, all right, so we're continuing now to discuss the signal transduction aspects of particularly fungi. And what we're going to do first, and you'll get to see better examples of this when we move on to some of the PowerPoint slides later. But what we're going to talk about is this phenomenon that occurs in a number of fungi. And that is that under certain circumstances, they can switch from either a yeast-like form to a filamentous form, or if they're normally filamentous, they can switch from a filamentous form to a yeast-like form. And for those of you that are not interested in fungi, but are interested in medicine, you may develop an appreciation for them because we'll talk about examples where that particular transition is important for particular diseases. We won't focus on that right now, but when we get to the PowerPoint slides, I'll show you examples. But the main thing is that, as we talked about before, both Saccharomyces and Usilago normally grow by budding. At least the haploid cells grow by budding. We did say that Usilago produces this filamentous dikaryon, but that's only during the process of mating and moving into that program of pathogenic development. For the longest time, people didn't realize that, or they forgot that yeast was a fungus. I mean, most people, when they think about fungi, they think about, you know, fuzzy stuff that grows on your bread. You're used to thinking about filamentous forms or organisms. And for a long time, people forgot that yeast is that way. And part of the reason for that is that over the course of years of using Saccharomyces as a genetic model, they essentially selected for strains that were able to grow as yeast-like cells because they form nice discrete colonies on petri dishes, which, if you're used to doing genetics with bacteria, is great because there's all sorts of things you can do with organisms that form nice colonies on a petri dish. But you know, for 50 years or so, that's what people work with when they work with yeast, with Saccharomyces. And so they forgot that in the wild, and if you're not working with lab strains, yeast actually do produce filaments, and it depends on their growth conditions. So typically, yeast will produce filaments instead of, well, actually what they do is they produce something called a pseudohyphal structure, where the mother-daughter cell buds, they don't separate. If you recall, I'll just draw this and then I'll erase it. If you recall, the yeast cell forms a bud, and then those separate. But what happens is, under these certain conditions of growth, or certain conditions of nutrient availability, instead of separating, what these guys do is they will continue to form a chain of budded cells. And this is what we refer to as pseudohyphae. And that is something that can take place with diploid cells or with haploid cells. With diploid cells, it looks more like this. With haploid cells, they form structures like this, but what happens is they penetrate the agar, so they become invasive. And this particular change happens when there is abundant fermentable carbon, i.e. like glucose, but there is low nitrogen. That's when you get pseudohyphal growth. And so what we're going to look at is, what are the signaling pathways that allow that particular change to occur? First, we'll look at that in Saccharomyces cerevisiae, and then we'll move on and look at that in Usilago matus and compare the two. Okay, and that will be the focus of this particular video. After that, we'll take a break, and then we'll move on to additional discussions that relate to signal transduction. but those would be using PowerPoint slides. Okay? All right, so far so good? All right. So remember, last time we talked about the signaling pathway that was used for the mating pathway. Assuming that this is still in the frame, just as a reminder, there was a sterol 20, MAP kinase, 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 which phosphorylates sterol 11, the MAP kinase, 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 which phosphorylates sterol 7, the MAP kinase, kinase, which phosphorylates Fuz3, a MAP kinase that is specific for this mating pathway by phosphorylating sterol 12, the transcription factor that activates Fuz1 for mating. Now, what we said last time was this signaling pathway is complicated because a number of the players in that pathway, as we talked before earlier in the semester, if you guys are fans of Game of Thrones, this will be another opportunity for you to explore your ability to keep track of the different characters. But each of these characters we're talking about here is involved in several other subplots, if you want to use the same analogy. In that, sterol 20, sterol 11, sterol 7 are participating in a variety of other signaling pathways. And the first one the one that we're going to talk about specifically has to do with this filamentation pathway. And this filamentation pathway, the first one we're going to talk about that affects that is a starvation pathway. So that's the pathway illustrated right here. I know this is a very busy whiteboard at this point, but we're going to focus on SHO1, which is a receptor in the cell membrane that responds to conditions of starvation. And once activated by the realization that the cells are in a starvation condition, SHO1 somehow is involved in activating sterol 20. So here we have sterol 20 again. As a reminder, sterol 20 has a kinase domain and a crib domain, and the crib domain is involved in blocking the kinase domain so that sterol 20 doesn't autophosphorylate itself, except when needed. Okay. Now, just as we talked about before, CBC42 is the G protein, or a G protein, that's going to be involved. And in this case, a second small G protein called RAS2 is also going to participate in this process. So RAS2 is going to somehow be involved in activating CBC42, which then is going to interfere with the auto inhibition that sterol 20 exerts on itself. This is basically going to open up the kinase domain so that now sterol 20 again can phosphorylate itself. So now it becomes active. Once sterol 20 is phosphorylated, it becomes active, and then it is going to in turn, so this is this cascade of phosphorylation, it is in turn going to phosphorylate sterol 11. So now we have sterol 11 again in this new pathway. Sterol 11, remember, is the MAP kinase, kinase, kinase. And here we introduce a new component. In yeast, there are two proteins called BMH1 and BMH2. These are part of a group of proteins that are called 14-3-3 proteins. And I think they may originally have been identified in mammals, but I'm not sure. But their main thing that they do is they couple areas or proteins that have been phosphorylated. So in a sense, BMH1 and BMH2 are acting as a kind of a scaffold to work with this use of these MAP kinases. Okay? So they're going to notice that these proteins are phosphorylated and couple together proteins for the purpose of carrying out this particular pathway. Okay? Now, once sterol 11 is phosphorylated, it is then going to phosphorylate sterol 7, the MAP kinase, and once phosphorylated, it's going to phosphorylate KSS1, which is the MAP kinase for this particular pathway. And once that's active, it's going to do two things. One is, if you recall from the mating pathway, DIG1 and DIG2 have been blocking a transcription factor called sterol 12. When KSS1 gets phosphorylated, it blocks them. Okay, so it's going to block or inhibit DIG1 and DIG2. And what that means is that now KSS1 can phosphorylate sterol 12, and that's going to make sterol 12 active. Now, in this particular example, for filamentation, sterol 12 has to interact with a second protein called TEC1. And the binding of these proteins is going to be at a particular location. So sterol 12 is going to bind to a PRE. PRE, I don't know if I'm still in frame here. I hope that I am. Yep, okay. PRE, that stands for pheromone response element. So if you recall from several lectures ago, we talked about response elements that are particular for different kinds of sets of genes being activated. So anybody remember any other kinds of examples of a response element? Not in the last two lectures, but this would be like when we were talking about regulation strategies.
Heat shock.
Yeah, heat shock response elements, estrogen response elements. So these are pieces of DNA that get bound by particular regulatory proteins or regulatory transcription factors that allow for discrete sets of genes to be activated all at once. So whenever any of these genes have a particular response element, the proteins that activate those genes bind to those response elements, and then they get coordinatedly activated. Okay, so PRE stands for a pheromone response element. TEC1 binds to a TCS, which I guess is a sequence that TEC1 likes to bind to. And then what do you suppose that an FRE is? What do you think an FRE is?
Well, the RE is response element.
Correct.
The F,
What pathway are we talking about now?
filament.
Yeah, that's right. So this is a filamentation response element. Okay, and so again, just to restate this, through this cascade, ultimately sterol 12 is phosphorylated and activated, and its inhibitors have been inhibited, and it's able to bind to the pheromone response element next to TEC1, who's binding to TCS, and both of those are part of what we call a filamentation response element. Now, what's the purpose of that? The purpose of that is to activate transcription of a gene called FLO11. And FLO11 encodes something called flocculin. And normally when you grow yeast, they stay suspended. As budding cells, they stay suspended in your culture. But when they express flocculin, they clump. So they flocculate. And so in like a big vat of yeast, what would happen is if they were expressing flocculin, they would kind of clump and then fall out of suspension. Okay? And if you think about it, that's what's going on, is it's basically what's happening is when flocculin is expressed, the mothers don't want to let go of their daughters. Now, some of you may have experienced that kind of thing where, you know, your parents don't want to let go, they get kind of clingy. Well, that's what's going on here, is the mother cells, they do not want to let go of their daughter cells. And so then, as each daughter's made and becomes a mother, it doesn't want to let go of its daughters, and that's how you get this chain that resembles a filament. Okay? Any questions about any of that pathway? Well, at the rate this is going, this may go a lot faster than I had anticipated. Okay, but that's only one way that we can get filamentation in Saccharomyces. There's another pathway, and this is going to make you kind of change gears in terms of your signal transduction brain, because now we're going to talk about the cyclic AMP-dependent protein kinase A pathway, which we introduced last time to make you not nervous about it, but we'll talk about it again just to remind you. Okay, so again, in a cyclic AMP-dependent protein kinase A pathway, which I should write that down somewhere. Oh, actually, before we do that, just before we do that, if you want to, if we can extend the view of the board, I'm just going to remind you of how this signaling pathway compares with what we saw for the mating pathway. Okay, so you can see that part. All right, very good. All right, so we just went through this pathway here today for the filamentation pathway. Notice that a lot of the same players were in the mating pathway. So we again had CDC42 activating sterol 20 then phosphorylated sterol 11 to activate it, which phosphorylates sterol 7, which in the case of the mating pathway activates Fuz3. Fuz3 also is involved in inhibiting DIG1 and DIG2 and activating sterol 12, but had the additional role of activating FAR1, which would lead to cell cycle arrest. So the cascade itself is very similar. The difference is, what's the signal that's causing that to happen? In the case of the mating pathway, you have pheromones interacting with the receptors in the membrane, and then that initiating a cascade that depends on the activation of a heterotrimeric G protein, which then leads to the rest of the steps over here. And if you compare that with what we're just talking about today, you see that the signal is different. So starvation is the signal, and the other thing is that we have changed up some of the players that are downstream from the signal. Now, in the case of the mating pathway, what is it that keeps the central components of the mating pathway together for the mating pathway? Yes?
Is it the STE5 scaffold?
That's right. So sterol 5 is a scaffold protein that holds these guys together so that they're aware that this is for the mating pathway, not for the starvation pathway. Okay? Now, there's one other thing I want to ask you guys while this is still up, and that is, what do you suppose STE stands for? You notice on this whole setup, you've got these proteins called STE proteins. STE2, STE3 for the receptors, STE20, 11, 7 for these kinases. Why do you think they're called STE or sterol 20, sterol 11? Think about what, go ahead.
Because if, like, the number might be where if there's like a mutation in like that certain like spot, then the yeast would be sterile.
Exactly, that's exactly right. So again, reminding ourselves that as geneticists, one of the first things we do is we isolate mutants and see how those affect particular phenotypes or outcomes. Any of these particular genes, or the genes for any of these components, if there were a mutation that inactivated those components, those cells would become sterile. They would be unable to mate. It just so happens that their name as a sterile, their name as a mutation that would cause sterility has been retained, even though in these cases, we're talking about a response to starvation, okay? So they were originally identified as mutations, as genes whose mutations would lead to sterility for Saccharomyces, okay? All right, any questions on any of that before we continue with the cyclic AMP-dependent protein kinase A pathway? Okay, I'm gonna, since we can see the whole board right now, I'm gonna write that down again, that name, and then we're gonna focus in on the other side of the board in just a second. So cyclic AMP dependent protein kinase A, otherwise known as PKA pathway. And just remember, I'm writing that over here, but it has nothing to do with any of this MAP kinase stuff. It's just the name of the pathway that we are about to discuss in detail. Okay? We'll focus on it over here now. Okay, so now we're going to continue to talk about the protein kinase A pathway. And as a reminder of just the components first, before we talk about the details of how they work. So in the protein kinase A pathway, it's dependent on cyclic AMP. Cyclic AMP is produced by an enzyme called adenylate cyclase. Okay? So adenylate cyclase makes cyclic AMP, and that's important because cyclic AMP is going to have an effect on the regulatory component of PKA. So there are regulatory subunits of PKA, and then there are also catalytic subunits of PKA. And normally, the regulatory subunit is bound and blocks the catalytic subunits. But when cyclic AMP binds in a spot on the regulatory subunit, that causes the regulatory subunit to be removed from the catalytic subunit, and that makes the catalytic subunit active. Okay, does that make sense? All right, so now we'll talk about the specifics of this pathway in Saccharomyces. So in Saccharomyces, you've got an alpha subunit G protein, so this is part of the heterotrimeric G protein complex, that when activated becomes independent. And when it's independent, it's now able to be used to respond to availability of glucose. So there's a receptor in the membrane called GPR1, which you can see here. And GPR1 responds to the levels of glucose. When there's sufficient levels of glucose, then the alpha subunit that's active will activate adenylate cyclase, illustrated here as AC. And adenylate cyclase will in turn make cyclic AMP. The regulatory subunit for Saccharomyces cerevisiae is called BCY1. Okay? And the reason for that is BCY stands for bypass of cyclase, and we'll see what that means later when we talk about actually mucilago. So the regulatory subunit, BCY1, normally has been binding to and blocking catalytic subunits, and there are 3 of them in Saccharomyces, just to make it easier for you to, in case you're having too easy of a time, now there are even more of them to remember. So the PKA catalytic subunits are referred to as TPK1, TPK2, and TPK3. And normally they're blocked by the regulatory subunit called BCY1. But once this pathway is activated and cyclic AMP levels are up, cyclic AMP will interact with the regulatory subunits, relieving the regulation on the catalytic subunits. And then the catalytic subunits then can do what they do, okay? Now, TPK2, what it's going to do is it's going to activate a transcription factor called FLO8. And FLO8 is going to bind and activate transcription of FLO11. And what did we say flow 11 was there for?
To make flocculin.
Yeah, to make flocculin. So this is essentially two different ways to activate the flocculin gene, and then that's going to lead to pseudohyphal growth. Okay? But meanwhile, you've got TPK1 and TPK3, and these guys normally are not involved in that process. They're more involved in a different feedback regulation. And when I say feedback regulation, what I mean is TPK1 and TPK3 are going to be involved in reducing the levels of cyclic AMP. And the way that I think this is happening, the way that I'm pretty sure the evidence supports, is the TPK1 and TPK3 phosphorylate two enzymes called PDE1 and PDE2. PDE stands for phosphodiesterase, and that's going to essentially act on cyclic AMP to reduce levels of cyclic AMP. So why would you want to reduce levels of cyclic AMP? I mean, if you have them around and they're activating this pathway, why would you ever reduce the levels?
If you were no longer in your starvation condition.
Exactly. So if you're no longer in the conditions that warrant this process and that warrant the production of flocculin to produce pseudohyphal growth, then it would make sense that you would want to be able to stop the signaling that allows this pathway to happen. Now, some of you have probably heard of this concept of second messenger. Have you? Yeah. Okay, so second messenger is this, and that is that the signal is the level of glucose. That's the signal from outside the cell. But the second messenger that allows that information to be transmitted, in this case, is cyclic AMP. So the levels of cyclic AMP should be reflective of how much glucose there is outside. And so we're constantly getting rid of cyclic AMP so that we have to continue to make it if the conditions outside are the same that originally warranted us activating this pathway. Okay? And so that's why TPK1 and TPK3 are involved in making sure that these phosphodiesterases, PDE1 and PDE2, lower the levels of cyclic AMP, so there has to be a constant signal from outside to keep that pathway going. Okay, questions? Okay, I know that's a lot of characters to keep track of, and we're about to introduce some more.